Welcome back to the deep dive. This is your shortcut, really, for getting your head around complex topics and making them stick. Today, we're taking a deep dive into something just incredibly fundamental for biology, for medicine, for you, protein metabolism. And look, this isn't just about, memorizing pathways from a textbook. It's about understanding these really dynamic processes, stuff that defines life, health, disease. Think of this as your fast track, designed for you, maybe a first-year med student, to clarify this. Intricate dance of proteins, connect the dots, and really anchor the clinical side with things you'll actually remember. Because proteins, they're everywhere, doing everything right. Building blocks, sure, but also enzymes, driving reactions, hormones. They make up, what, 15–16% of our body weight? That's huge. So understanding how our body manages them, builds them, breaks them down, deals with the waste that's not just academic. It's
Yeah.
central to patient care, isn't it?
Oh, absolutely. It's the bedrock for so much. And yeah, this deep dive will guide you through that whole journey. We'll start with the basics, the Lego blocks, amino acids, and how they're processed. Then we'll tackle how the body cleverly handles the toxic nitrogen waste. That's a big one. And maybe most importantly, we'll look at what happens when these systems go wrong, those metabolic disorders you will see. Understanding the why is really key.
Okay, so let's zoom out first. Proteins are vital. We get that. But what exactly are they? What makes them so indispensable?
Well, think of them like these incredibly versatile molecular machines. And they're built from smaller units, the amino acids. There are 20 different kinds, and each one has this unique side chain, giving it a sort of different chemical personality, a different job it can do. Now, what's really critical right from the start is this. Your body can make some of them the non-essential ones, but 9 of them, the 9 essential amino acids, your body just cannot synthesize. You absolutely have to get them from your diet, period. And for you, heading into medicine, remembering those 9, it's non-negotiable. So here's a mnemonic, kind of an anchor for you. If learned this, huge list may prove truly valuable.
Okay, let's hear it.
So that's isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine, and histidine. The huge list may prove truly valuable.
Got it. That's helpful.
And then there are also these conditionally essential ones like arginine, tyrosine, cysteine. They become essential during times of high demand. Think kids growing fast, pregnancy when the body's own production just can't keep up.
That mnemonic is great, thank you. And it really highlights this constant need, this flow of amino acids, which brings us to nitrogen balance, right? You mentioned it, the body's metabolic scale of life. How does that work?
Yeah, it's a really elegant concept. Basically, we look at 3 states. First, most healthy adults, they're in zero nitrogen balance. Equilibrium, what goes in terms of nitrogen from protein equals what goes out, mainly is urea. No net change in body protein. Simple enough.
Okay.
Then you have positive nitrogen balance. Intake is greater than output. This is your building up state. Totally normal, even desirable, during growth spurts, pregnancy, or when a patient is recovering from malnutrition or a major illness. They're rebuilding tissue. And clinically, you see this in pregnancy, right?
Yeah.
Fetal growth, maternal tissue expansion, plus hormonal changes actually suppress some urea cycle enzymes a bit, saving nitrogen for building.
Makes sense.
But interestingly, positive balance can sometimes be abnormal, like in hypothyroidism. Metabolism slows down, so amino acid breakdown might decrease. There's not healthy growth, it's sluggishness.
And the third state, the one we worry about, negative nitrogen balance.
Exactly. Negative balance intake is less than output. This is almost never good news. It means the body is breaking down.
Its own proteins.
Yes, breaking down its own reserves.
Yeah.
You see this dramatically after major trauma, severe infections, bad burns, or even just prolonged starvation. There's massive breakdown, catabolism of muscle protein. Stress hormones like cortisol, adrenaline, they flood the system, driving that breakdown to get amino acids for energy or for making acute phase proteins. The scale tips hard towards breaking down.
That really paints a picture of how dynamic things are and how the body prioritizes under stress, this constant building and breaking down.
That's protein turnover, right? Happening all
All the time. We're talking 300, maybe 400 grams of protein turnover every day. It's incredible. Some proteins last minutes, others for weeks. Just constant renewal. And all this activity feeds into this central hub, the amino acid pool.
The pool. Yeah, it's actually pretty small, maybe 100 grams or so in an average adult male, but it's incredibly active. This is where dietary amino acids, ones from body protein breakdown, and any newly synthesized non-essential ones all mix together. And from that pool, they get whisked away, build new proteins, make other nitrogen compounds, or if needed, their carbon skeletons get burned for energy.
Okay, let's unpack that a bit more. Proteins break down, amino acids hit the pool. What next? How does the body process them, especially if they're being used for energy? This feels like where the nitrogen disposal challenge comes in.
You got it. That's the crux. So protein catabolism, the breakdown, happens in stages. Stage 1 is extracellular digestion, your gut.
Right. Chewing, stomach acid.
Exactly. Mechanical breakdown, then HCl in the stomach unfolds them, pepsin starts chopping. Then in the small intestine, pancreatic enzymes like trypsin, chymotrypsin go to work. Finally, brush border enzymes finish the job, giving you single amino acids, small peptides, ready for absorption. But stage 2, this is where the nitrogen issue really hits home. Intracellular breakdown, mainly in the liver. This is where that amino group, the NHROs, has to be removed.
Why? What's the big deal with the amino group?
Because once you detach that nitrogen, the remaining carbon part, the keto acid, is useful. Go into energy pathways, be made into glucose, whatever. But the nitrogen itself, that needs careful handling.
Okay, so removing the nitrogen is key. And there are two main ways the body does this, transamination and deamination.
Spot on. Think of transamination first. It's like an amino acid swap meet. It helps gather nitrogen centrally. So an amino group from one amino acid gets transferred onto a keto acid. Often it's alpha-ketoglutarate. That alpha-ketoglutarate then becomes a new amino acid, glutamate. And the original amino acid becomes a keto acid.
So it swaps partners.
Precisely. It's a way to funnel amino groups onto glutamate, which is then easier to deal with. And the enzymes doing this swap. They're called aminotransferases or transaminases. Crucially, they need a helper molecule, pyridoxal phosphate.
Which comes from?
Vitamin B6. So B6 is absolutely vital here. It's the amino group shuttle. If you're low on B6, this whole process can get sluggish.
And these aminotransferases, they're pretty famous in clinical labs, aren't they?
Oh, very famous. This is where you meet ALT alanine aminotransferase and AST aspartate aminotransferase. These are basically your liver's report card.
Right. The liver function tests, LFTs.
Exactly. When liver cells get damaged, think viral hepatitis, drug toxicity, alcohol abuse, these enzymes leak out into the blood. So high ALT and AST levels, that signals liver trouble. And a little clinical pearl for you. In alcohol-related liver disease, the AST is often much higher than ALT, usually a ratio greater than 2. That pattern can be a useful clue. The key insight is why they're markers. They belong inside healthy liver cells. Seeing them in the blood means cells are breaking open.
Okay, so transamination collects the nitrogen onto glutamate. Smart. But sometimes you need to actually get rid of the nitrogen, right? Not just move it around. That's deamination?
That's deamination, the direct ammonia release. Here, the amino group is snipped off as free ammonia, NHESs. This happens mainly in the liver and kidneys. You've got enzymes like amino acid oxidases, glutamate acting on glutamine, and a really key one, glutamate dehydrogenase, which takes that glutamate we collected and directly releases its amino group as ammonia. Now, here's where it gets really interesting and, frankly, quite critical for understanding why the body goes to such lengths. I want you to think about this. Why is free ammonia such a big deal? What makes it so toxic, especially to the brain, that we have these complex, energy-hungry systems just to handle it? We'll come back and nail that down.
Okay, so let's tackle that question head on. Why all the fuss about ammonia? Why is it the brain bully? It's toxic for several really nasty reasons. Your blood ammonia has to be kept incredibly low, like 25 to 40 micromoles per liter, tiny amounts, because if it gets higher, it wreaks havoc on the brain, which is super vulnerable. How does it do that?
Well, first, it messes directly with the TCA cycle, your cell's main energy generator. Ammonia reacts with alpha-ketoglutarate, a key player in that cycle.
Pulling it out of the cycle.
Exactly. It depletes alpha-ketoglutarate, essentially choking the TCA cycle and starving brain cells of ATP, of energy. An energy-starved brain is a malfunctioning brain. Simple as that. But it doesn't stop there. Ammonia also interferes with amino acid transport into the brain, messes with protein synthesis, screws up the pH balance, affects blood flow, makes the blood-brain barrier leaky. And it disrupts neurotransmitter metabolism, glutamate, GABA. Basically, it throws a wrench into almost everything the brain needs to function properly. Plus, it causes osmotic problems, leading to brain swelling cerebral edema. That's a life-threatening emergency. The brain's high energy needs make it uniquely susceptible.
Okay, that paints a very clear, very scary picture of why ammonia is bad news. So if it's made in places like muscles during amino acid breakdown, how does the body safely get it to the liver for detoxification? It can't just float free. It needs a chaperone, right?
It absolutely needs ammonia chauffeurs. And the body uses two main ones, both non-toxic, alanine and glutamine. Alanine works via the glucose-alanine cycle. This is mainly muscle sending nitrogen to the liver. In muscle, pyruvate grabs an amino group, becomes alanine. Alanine travels in the blood to the liver. In the liver, alanine gives back the amino group, which becomes ammonia for the urea cycle, and turns back into pyruvate, which the liver can use maybe to make glucose. It's a neat two-way street.
Yeah.
But the major carrier is glutamine. It's made by the enzyme glutamine synthetase, which basically sticks ammonia onto glutamate.
Mopping up the ammonia.
Exactly. Glutamine is harmless. It's the most abundant amino acid in your blood, and it carries ammonia safely to the liver and also to the kidneys. In the liver, an enzyme called glutaminase chops the ammonia back off, feeding it into the urea cycle. In the kidney, glutamase also releases ammonia, but there it can be excreted directly into the urine, which is important for managing acid-base balance.
So the chauffeurs drop off the ammonia at the liver's doorstep, and that's where the main event happens. The liver's detoxification plant, the urea cycle, also called the ornithine cycle. This is the big fix.
This is the big fix. Its whole purpose: convert that super toxic ammonia into something non-toxic, easy to get rid of, and packed with nitrogen, urea. This multi-step pathway happens only in the liver. And interestingly, the first couple of steps are inside the mitochondria, the rest out in the cytosol. Shows how compartmentalized things are. You need ammonia, bicarbonate, and also aspartate that brings in the second nitrogen atom for urea. And the end product, urea. It's brilliant, high nitrogen content, super water soluble, totally inert metabolically, perfect for peeing out. And here's a crucial point about regulation. Eat a lot of protein. Your body upregulates the urea cycle enzymes. Makes sense, right? More protein breakdown, need more detox capacity.
And the opposite, low protein or starvation.
It downregulates them. Saves energy, less nitrogen load anyway. But this regulation is key clinically. Think about refeeding syndrome.
Right, you mentioned that.
Yeah. You take someone who's been severely malnourished. Their urea cycle enzymes are running low. If you suddenly blast them with high-protein feeds,
Their system can't cope.
exactly, the ammonia load overwhelms the sluggish urea cycle. You get hyperammonemia and boom, all those toxic brain effects we just talked about. It's why refeeding has to be done carefully, gradually. The urea cycle is the brain's crucial safety valve against nitrogen toxicity.
We've painted this picture of really sophisticated machinery, but inevitably sometimes things break. A missing enzyme, a faulty gear. This is where we see the real clinical impact, right? These metabolic disorders.
Absolutely, and there are quite a few, actually, over 50 known inherited disorders of amino acid metabolism. They're individually rare, but add them up and they're significant. Usually it's a faulty enzyme, maybe partially active, maybe completely gone. And result, toxic buildup of an amino acid or its byproducts. And sadly, if they're not caught and treated early, severe intellectual disability is a common outcome, which is why that heel prick test on newborns is so incredibly important. Screening for things like PKU, homocystinuria, it's literally life-changing, mind-saving.
Okay, let's drill down on two key ones every med student needs to know. First up, phenylketonuria, PKU.
Right, PKU, the classic phenylalanine toxic traffic jam. It's the most common inherited amino acid disorder, maybe 1 in 15,000 births. The problem is a missing or faulty enzyme, phenylalanine hydroxylase, or PAH. Its job is to convert phenylalanine into tyrosine.
So without pH?
Phenylalanine just piles up. In the blood, in tissues. The body tries to get rid of it through other routes, making phenylketones like phenylacetate. These spill into the urine, giving it this characteristic musty, mousy smell. But the real damage, that buildup, especially phenylpyruvate, actually poisons mitochondria by blocking pyruvate uptake. It messes up brain energy metabolism badly.
Leading to the symptoms?
Exactly. Severe intellectual disability, seizures, small head size, microcephaly, delayed development, even lighter skin and hair, because tyrosine production, needed for melanin, is also affected. Treatment is lifelong and strict. A very low phenylalanine diet. You have to supplement with tyrosine. And absolutely avoid artificial sweeteners like aspartame, which contains phenylalanine. Early diagnosis and diet make a huge difference.
Such a clear example of one broken step causing system-wide chaos. Okay, what about the other one you mentioned, homocystinuria, HCU?
HCU. I call it homocystine's double trouble. It's rarer than PKU, maybe 1 in 344,000. Most often, it's a deficiency in cystathionine synthase, CBS. This enzyme normally converts homocysteine into cystathionine, sort of the next step down the pathway. It can also be a problem with methionine synthase.
So homocysteine builds up instead?
Yep. Homocysteine and its precursor, methionine, accumulate in the blood. You find oxidized homocysteine, in the urine. Clinically, it's tricky. Patients often have dislocated lenses in their eyes. They can be tall with long fingers, what we call a marfanoid habitus, and often have intellectual disability.
Sounds a bit like Marfan syndrome.
It does, but the underlying cause is different. Marfan's is a defect in the fibrillin-1 gene itself. In HCU, the high homocysteine seems to mess up the structure and cross-linking of fibrillin. Similar look, different metabolic root cause. Needs careful diagnosis. Treatment involves a low-methionine diet and supplementing with cysteine, which becomes essential, plus high doses of vitamin B6, B12, and folate, all cofactors involved in homocysteine pathways.
And briefly, what about urea cycle disorders as a group?
Right, these are genetic defects in any one of the 5 core enzymes of the urea cycle itself. As you'd expect, the main consequence is hyperammonemia, high ammonia levels, because the detox pathway is broken. You also get buildup of whatever intermediate comes just before the faulty enzyme. OTC deficiency, ornithine transcarbamylase deficiency, is the most common type.
And symptoms related to the high ammonia.
Absolutely. They vary a lot depending on how bad the enzyme defect is, but especially in newborns, you can see vomiting, extreme lethargy, irritability, poor feeding, progressing to seizures, coma, developmental delay if they survive. It's serious. Treatment is tough. Strict low-protein diet is essential. Sometimes they use drugs to help remove ammonia or replace amino acids with their keto acid versions to try and soak up some nitrogen.
Okay, beyond these major pathways and diseases, are there other specific markers in protein metabolism we might see day to day in the clinic?
Yes, definitely. A really important one is creatinine. Think of it as the muscle meter and kidney compass.
Oh.
Creatinine comes from the breakdown of creatine and creatine phosphate, which are almost entirely found in muscle tissue. It's produced pretty constantly, and the amount produced is proportional to your muscle mass.
So you can estimate muscle mass from it?
You can, from a 24-hour urine collection. But its main clinical use, the one you'll see constantly, is as a marker of renal function, kidney function.
How so?
Because healthy kidneys filter creatinine out of the blood very effectively. If your blood creatinine level starts to rise, it's a strong sign that your kidney filtration is impaired, that nephrons are damaged. It's a fundamental kidney test.
That's a really practical one to remember. Okay, one last thing. Let's circle back quickly to mobilization of protein reserves, when the body really needs to tap into its own protein.
Right. This happens under extreme stress starvation, major injury, severe illness, and it's tightly controlled by hormones. You have anabolic hormones, insulin,
Right.
growth hormone pushing towards building protein, reducing breakdown. Then you have catabolic hormones, especially glucocorticoids like cortisol.
Cortisol increases breakdown.
Increases breakdown, decreases synthesis. It shifts the balance towards liberating amino acids, maybe for energy, gluconeogenesis, or making other essential molecules. And a really clear clinical example of too much cortisol-driven breakdown is Cushing's syndrome. Patients have chronically high cortisol, which leads to excessive protein breakdown, particularly collagen in the skin. This weakens the skin, causing those characteristic purple stretch marks, or striae. It's visible protein catabolism.
Wow, what a journey. We've gone from the basic roles of proteins and amino acids all the way through nitrogen handling, that crucial urea cycle, and then into what happens when it goes wrong, PKU, HCU, urea cycle defects, even seeing the effects in things like Cushing's or kidney function tests. It's complex, but it all connects.
It really does. It's this incredibly elegant, interconnected system. And the insight, I think, for you is seeing how it's connected. Understanding the normal function makes the abnormal states, the diseases, make so much more sense. And that's absolutely vital for diagnosing and treating patients effectively down the road, gives you a solid framework.
So wrapping this up, what does this all mean for you listening?
Well, it's a stark reminder of this constant balancing act our bodies perform, this tightrope walk between building up and breaking down protein, and how quickly things can shift under stress or due to disease with potentially devastating consequences if we don't understand the underlying metabolism.
And maybe a final thought to chew on, something a bit provocative. Think beyond just diet. How might other factors, like chronic stress, which we know affects cortisol, how might that subtly nudge your own nitrogen balance, your protein turnover, even without any obvious illness? What everyday choices, perhaps seemingly small ones related to stress management or lifestyle, might actually be influencing this fundamental metabolic scale, tipping it slightly towards breakdown or building?
A really fascinating question to leave us with. Thank you so much for joining us on this deep dive into the absolutely vital world of protein metabolism. We really hope this has clarified things and given you some solid anchors for the future.